Hello and welcome to another video session. In this video, I'm going to talk about the details of connective tissues. So let's begin. Connective tissues. What are some of the connective tissues that we have in our body? Now, the connective tissues that we have are tendons. We can talk about ligaments. We can talk about what we call the connective tissue proper. We can talk about the blood. Blood itself is a connective tissue. Bone is also a connective tissue. We'll be talking about different types of the connective tissue proper very soon. And then we also have adipose tissue, which is also a connective tissue, fat tissue, adipose. So these are some of the examples of connective tissue. Now, what's the origin of connective tissue? Where does it originate? During the embryological development, it originates from the structure that we call the mesoderm. Mesoderm, right? So during the embryological cycle, you have three layers that occur. The outer layer, which is called the ectoderm, right? Then the inner layer of tissue, let's do this. So this is called the mesoderm. The middle layer is called the mesoderm. So this is a part that gives rise to the blood-forming tissues, the spleen, liver, thymus, cartilages, and so on. The muscles, they all come from mesoderm. Ectoderm has to do with structures on the surface of the skin. Then you have the endoderm, the inner tissue or inner layer is the endoderm. Right. That gives rise to some of the internal structures. Okay, so we're talking about the mesoderm over here. That gives rise to the connective tissue. Right. Now let me also add here cartilage. Cartilage is also another type of connective tissue. Okay. All right. So we'll be talking about the different components of the typical connective tissue. And I just want to make mention of the fact that there will be different variations in the amount of the elements that we find in the connective tissue. So we can have something like a dense connective tissue, which occurs in structures such as the ligaments, tendons, aponeurosis, fascia, fibrous capsules, and so on, right? So we can make mention of a dense connective tissue. How dense it is based on the type of fibers that you see in there. So dense connective tissue. So we're going to say that this occurs in structures such as ligaments. So found in ligaments, tendons, then what we call the aponeurosis. Okay. Fascia, fibrous capsules, and so on. Now, this is because you have a large amount of collagen fibers in here, all right? So it has large amounts of what we call the collagen fibers. So that makes it very dense, and they have this tightly packed fibers. So the fibers are tightly packed, so dense. Then the second one will be the loose connective tissue, loose connective tissue. Now, the loose connective tissues, on the other hand, contains relatively few collagen fibers. So this tends to have more ground substance and tissue fluid. Right? So connective tissue, loose connective tissue have relatively few collagen fibers. But more ground substance. And tissue fluids. And then we can have the fatty tissue. So fatty connective tissue. So these type of tissue, connective tissues have a large accumulation of fat cells. So they have accumulation of large fat cells. Okay. So those are the adipose tissues. So the three varieties of connective tissues that we just mentioned are the dense connective tissue, loose connective tissue, and fatty connective tissues. So those are the typical connective tissues that usually you come across. But there are intermediate gradations between these three varieties. And also there are numerous other varieties that have also been identified. So in some regions, for example, if you take the ligamental flavor, there's a marked increase in the number of elastic fibers at the expense of other components. An important function of the so-called connective tissue is frequently not to connect, but rather to separate, right? The capsule of many glands, for instance, consists of a fibrous connective tissue which effectively isolates the gland constituents from neighboring structures and enables them to function with minimum disturbance. So hence, for example, though the liver and the kidney tissue come into close contact, their fibrous capsule effectively intervene between the liver cells on one hand, and then the renal glomeruli and tubules on the other hand. So there's minimal disturbance between the organs. Okay. So it's about separation of the organs for minimal disturbance. So let's now look at the constituent element of connective tissue. So what are constituent elements of connective tissue? The first one I want to talk about is the fiber, or let's talk about the fibers. The fibers. So that's the American way. If you're thinking of the British way, then the British will write, we spell the fibers like this. Okay. So depending on which part of the world you are, you spell it differently. So fiber or fibers, our connective tissues generally consist of three main types of fibers. So there are three main types. So you can have collagen fibers. You could have the elastic fibers, or you could have the reticular fibers. So these are the types of fibers. So collagen, elastic, and reticular fibers. Let's narrow down to the collagen fibers. So what are collagen fibers? So collagen is a white, insoluble fibrous protein, which when treated with water gradually breaks down to form soluble gelatin or glue. So collagen, this is white and insoluble fibrous protein. So it's a protein, fibrous protein. Now, when you put this in water, it breaks down to gelatin, soluble gelatin or glue. So it breaks down. Okay. So collagen fibers are present in all connective tissues. Every connective tissue you take, they are present in all connective tissues. Including bone and cartilage. They usually possess a low refractive index, which in certain situations, for example, the hyaline cartilage happens to be the same as that of the ground substance which surrounds them. So they are difficult to see, very difficult to see them, because of the refractive index, how much light they're able to refract. Okay. Now, in dense connective tissue, the collagen bundles are much more numerous and more closely packed than in the loose connective tissue, as I've said before. So the dense connective tissue, here you have closely packed collagen fibers. Whilst in the loose type, you have like they're loosely packed. Okay. If you take the tendons, you realize that the collagen bundles run parallel with one another, right? So the tendons, take the tendons for example, you see that the collagen fibers are running parallel to each other. Okay. Now, collagen fibers have very high tensile strength. So they're very strong. Okay, when you stretch it, it's difficult to break them apart. But there's a limit to the tensile strength, because there's a limit. Once it reaches a limit, then they can break rather than stretch. That's why someone has a sudden strain, the fibers can break apart. Right. So because of the high tensile strength, that's why they appear in tendons, they appear in ligaments, they appear in the aponeurosis, the fascia and so on. If you take the abdomen, you have fascia in the aponeurosis, they appear there because of the strength. the fibrous pericardium, right around the heart. It also prevents sudden overextension of the heart, whilst the fibrous tunica adventitia performs a similar function for the large blood vessels, right? So the presence of collagen fibers over here are very useful. Some of the ways in which this content varies is in response to the functional needs. And they are very well illustrated in the structure of the skeletal muscle. Within the muscle itself, the connective tissue, both of the endomysium and the perimysium, contains relatively few collagen bundles and is particularly loose in texture, thus permitting free and unimpeded movement of the muscle fibers and also adjacent fasciculi, upon one another. It is because of this especially loose interfascicular connective tissues that muscles fasciculi can be so easily separated in the so-called muscle splitting operations, right? So that allows the nature of the arrangement allows muscles, the fasciculi or the muscles, to be split apart. When you do operations, you can easily separate the fibers because of the loose interfascicular connective tissue, allow for separation and working on the individual muscle fasciculi. Now, the epimysium, however, may consist either of loose connective tissue or be extremely dense, right? In muscles where the entire muscle belly is free to move, the epimysium consists of a loose connective tissue, which contains a relatively large amount of fluid in its interstices and really functions as a bezier, although it does not have the structure of one. Okay. Now, the collagen is formed as a result of the activity of one of the connective tissue cells, the fibroblast. So this collagen is formed from fibroblasts. Right, so that's the cell that produces the collagen. And we need vitamin C is very important in formation of collagen. So vitamin C is needed. Yeah. So that's why when it comes to wound healing, vitamin C is very important. It's essential. Now, there are certain things that can hinder the production of collagen fibers. So those that have the inhibitory effect on production. This includes the steroids, like the steroid hormones. Okay, that's why they can inhibit wound healing. Cortisol or the cortisone. So they can decrease the production of the collagen. So this is the overview of the collagen fibers. Now let's talk about the elastic fibers. So second one be elastic fibers. So the elastic fibers are single and they undergo branching so as to form a diffuse network. Yes, single and undergo branching. So they're branched. Okay, so you're going to see them maybe like this. You know. Now the network under varying degrees of stretch is to be considerable extent responsible for the maintenance of a slight tissue pressure, ranging from 2 to 14 centimeters of water. All right. So they have to maintain stretch due to tissue pressure. Now, if you take the superficial fascia of the elastic fibers in the skin, you see a lot of elastic tissue. You know, the elasticity of the skin is due to the presence of the elastic fibers. They are attached to the dermis and then epidermis of the skin. So the elasticity of the skin is due to the presence of the elastic fibers attached to the dermis and epidermis. So if it's in a young person, if you take a fold of the skin, when released, it snaps back into position almost at once. But in older people, it subsides much more slowly because older people have less elastic fibers in their skin compared to the young ones. Then the last type of fiber that we look at is the reticular fibers. Reticular fibers. So the reticular fibers form a thin branching network, not well marked in the subcutaneous connective tissue, right? So if you look at subcutaneous connective tissue, it's not well marked. So they form a thin branching network. A thin branching network. And you see reticular fibers mainly in the lymph nodes. So found in the lymph nodes and other blood forming organs. Now it's believed that usually they form the preliminary stage in the formation of collagen fibers, and they are effectively demonstrated by the silver impregnation test. All right, so you can use a silver impregnation test to detect them, especially when collagen fibers are formed. So they are probably right. So that is the overview of the reticular fibers. You can demonstrate them using the silver impregnation test. Now, so these are the fibers. We've spoken about 3 types of fibers: the collagen fibers, elastic fibers, and then the reticular fibers. Now, what are the cells that are found in the connective tissues? There are cells found in the connective tissues. So cells. Now, the cells that are found in the connective tissues are: 1, we have fibroblasts; 2, we have macrophages. So macrophages. 3, you have the primitive mesenchymal cells. 4, you have the mast cells. 5, you have fat cells. You can have pigment cells. You can have plasma cells. And then we can also have the blood cells. So these are different types of cells that you can find in the connective tissue. Now let's talk a little bit about the details of each of these. So let's begin with the fibroblast. Fibroblast. So the fibroblasts usually occur close to collagen. So they are close to collagens. All right. Their nuclei are large and they are oval. So if you look at the fibroblast, take a fibroblast, you have oval, large oval nucleus. Large oval nucleus. And they contain relatively little chromatin, but one or two large nuclei. So if you take a nucleus, you have little chromatin. They may have one or two nucleoli. And the cytoplasm don't have granules in them, and they may pass into fine elongated branching processes, except during mitosis when the cell becomes rounded. Right, so cell becomes rounded. So during mitosis, the cell becomes like rounded. Yeah. So, and then they also develop the processes that I just mentioned. So extensions of the cell membrane. Okay. Now, the chief function of the fibroblasts appears to be the formation of collagen, which first forms around them, right? So you see the collagen forms around it, becomes like you see this homogeneous material around the fibroblast, right? Which represents the collagen. So the main role is to produce collagen. Produce collagen fibers. Now, the collagen fibers, once they are produced, you see that they have closely packed transverse striations on them. Right? The newly formed fibers become oriented along lines of tension. That's the orientation, okay, along lines of tension. So fibroblasts play an important role in the healing of surgical incisions and other wounds, right? So play a role in wound healing. Very important in wound healing. Right? So if there's somebody gets a wound, what happens is that there's a surgical incision, right? The gap is first filled with a clot of blood or plasma. which is then invaded by blood capillaries and connective tissue cells. So among these cells, the macrophages act as scavengers, and then they remove all the debris, while the fibroblasts lay down the collagen fibers, which afford a firm and lasting union. So that's what happens when there's an incision, right, or there's a wound. Okay. So during the early stages of the healing process, the tissue is highly cellular and it has a rich blood supply. So as the amount of collagen increases, the number of fibroblasts and of the blood cells also diminish. All right. So that finally there remains a relatively avascular mass of collagen fibers, which is pale in color, and that constitutes the scar. Yeah. Now, fibroblasts are not phagocytic, and they show little affinity for vital dyes used in the lab, right? Though they may occasionally contain small droplets of fat. Okay. So that is what the fibroblasts, that's what they do. Main function is to lay down collagen fibers. Now, what about the macrophages? The macrophages. Now, the macrophages are present in loose connective tissue in about the same numbers as the fibroblast, right? Although they are subject to considerable variations. So we see this in loose connective tissues, present in loose connective tissues. If you look at the nucleus or the macrophage, they are usually small and darker than that of the fibroblast, right? So you see them usually small and darker than the fibroblast. And the cell is often elongated. See them a little bit elongated most of the time. You know. Like this. So the living macrophage has a very active surface membrane. These cells are intensely phagocytic. So these cells are phagocytic. So they engulf particles, they take in particles like bacteria. So they're phagocytic. Now, the macrophages can pick up dye stains, such as the tryptophan blue. So they can store vital stains, such as tryptophan, sorry, the word is tryptophan, the tryptophan blue. So this is a type of dye used for staining in the lab. So the main function of macrophages is to, clear the site of infection, all right, because it's phagocytic. helps to fight off infection. Now let's go on to the function of the primitive mesenchymal cells. The primitive mesenchymal cells, what are the functions? These are scattered irregularly through the connective tissue, though they most frequently occur close to the walls of the blood capillaries. And they look like the fibroblasts. They look almost like fibroblasts. So they're scattered irregularly through the connective tissue. They are close to capillary walls. Now, they are sometimes called the adventitial cells, and they are believed to be persisting undifferentiated cells of the primitive mesenchyme. So if you take a mesenchyme, they are believed to be the undifferentiated cells of the mesenchyme. So also called adventitial cells. So they are persistent, undifferentiated cells of the primitive mesenchyme. Usually under normal conditions, they appear to be completely inert, right? But they are nevertheless thought to be capable of rapid growth and differentiation into macrophages and other cells in response to the appropriate stimuli. So if there's an appropriate stimuli, they become macrophages or other cells. That appropriate stimuli, they become macrophages or other cells. Okay. So that's a function of the primitive mesenchyme. Now, the next one we'll talk about are the mast cells. The mast cells. So the mast cells are filled with large granules. These are filled with large granules. And which stain metachromically. That means purple with a Tulidine blue. Stains metachromically. metachromatically. So when say something stains metachromatically, it means a tissue is said to stain metachromatically with a dye when the color of the stained tissue is different from that of the dye, right? So a color is different from that of the dye, then we say that it stains metachromatically. So in this case, it's with the purple. It's purple with toluidine blue. Okay. So the blue dye becomes purple. Now the mast cells often occur in groups close to blood capillaries. So occur in groups close to blood capillaries. Now, it is believed that among other substances, they secrete histamine and heparin. So the common chemical they release is histamine, secretes histamine, and heparin. Remember, histamine plays a role in allergic reactions, whilst heparin it plays a role in inhibiting blood clots. Now, the presence of an anticoagulant in a tissue is of importance in case fibrinogen should escape from the blood capillaries, right? So for the anticoagulant can then prevent clotting of the tissue fluid, right? So that's the purpose of having this in the tissue. The next one, we'll look at the fat cells. Fat cells. We can find small droplets of neutral fat in the fibroblast and other connective tissue cells, right? So we find small droplets. Of neutral fat. Now, but if you look at the true fat storing cells, the fat completely fills the cells so that the nucleus is flattened and lies close to the cell membrane. So in the true fat storing, what we call the fat storing cells. In true fat storing cells. The fat completely fills the cell. Okay, so what you have will be something that looks like this. the nucleus will push close to the cell membrane, and then the fat will fill up. All right, so the whole this will fat. Okay. Now, the fat cells usually tend to develop close to blood vessels and may occur singly or as accumulation, which constitute fat or adipose tissue, right? So you can have them close to blood vessels as single cells, or you can have accumulation of cells as in fatty or adipose tissues. All right, so in adipose tissues, it's accumulation of so many fatty cells. Okay, so if you look at the peritoneal tissue, the retroperitoneal tissue being the peritoneum, right? Like the greater, what we call the greater omentum and mesentery. You have a lot of fat tissues in there. You know, like the belly, that's where the belly fat and other things come in. Now, remember, there are different types of fats that you also come across. We have something called the so-called brown fat. Brown fat that has been shown to be present in the human unit neck, in the neck of the human unit, okay, between the scapula and around the great vessels. They may be responsible for heat production in the neonatal period. So here, the fat consists of several discrete globules separated by cytoplasmic partitions. If you look at the tissues in the brown fat I'm talking about. Okay, right. So let's move on to the pigment cells. Okay, what are the pigment cells? Now, these are small branch pigment cells found in the dermis, and they contain melanin. So small, and they are branched. So the dermis is the connective tissue under the skin. And they contain the pigment melanin. Now, it's not usually clear if they actually form the pigments or whether the cells are connective tissue phagocytes whose pigment is derived from the epidermis, right? Now, cells which form their own pigments called the melanoblasts are found in the dermis and also in the choroid coat of the eye. So apart from these sites, pigment cells are uncommon in the connective tissue in mammals. So example of pigment cell here will be the melanocytes. Melanoblast. They become the melanocytes. So this is an example of the cell in epidermis. Then we'll talk about the plasma cells. The plasma cells. You find a lot of this in the lymphoid tissue. But in ordinary connective tissue, they are not usually common. They possess a small, dense, eccentric nucleus and basophilic cytoplasm. So they have a small, dense, eccentric nucleus. They have basophilic cytoplasm. That means they stain with the basic stains. And they have a lot of closely packed concentric lamellae of endoplasmic reticulum. The function of the plasma cells is essentially to produce antibodies. So they play a role in antibody production. And then the last type of cell that we'll look at are the blood cells in the connective tissue. So what are the blood cells in the connective tissue? Now, even in healthy cell, various form cells or the blood pass in small numbers through the walls of intact blood capillaries, and they enter the connective tissue. So in it, red cells, neutrophilic and eosinophilic granules, lymphocytes, monocytes may be found, right? So you can find you can find red blood cells, You can find the white blood cells. So the neutrophils. You can find lymphocytes. You can find eosinophils. You can find monocytes. And you can find all the cells, even the healthy connective tissue. Now, during a disease state, however, then the migration of the blood cells into a connective tissue may be markedly stimulated. Thus, in many types of bacterial inflammation, there's a greatly increased accumulation of neutrophil granules in the connective tissue of the inflammatory focus. And these cells constitute an important means of dealing with many bacteria. Okay. And then in some types of bacteria and in many viral infections, there may be local accumulations of lymphocytes, right? This also occurs with grafts of foreign tissue, which are not compatible with their host. The process of graft rejection is to a large extent due to many lymphocytes which migrate to the grafted area from the bloodstream. Cells which enter the connective tissues from the bloodstream are said to be hematogenous, while those which are formed in the connective tissue are histogenous, right? So those are some words that we also want to make mention of here. Cells that enter the connective tissue from the bloodstream, we refer to them as hematogenous. So we have hematogenous, hemato, right? From means hemat is blood. So cells from the bloodstream to the cell, to the connective tissue. From the bloodstream to the connective tissue. And those that originate from within the connective tissue. So those formed in the connective tissue are histogenous. Histogenous cells. Cells formed in the connective tissue. So these are the cells and the components, other elements that you find in the connective tissue. This brings us to the end of the different elements or the structure of the connective tissue. I hope you find this useful in your studies. See you again in my other videos.